face a complex epidemiologic landscape, seeing signs of autoimmune-mediated neurologic disease, thrombotic, thrombocytopenic purpura. This is, I believe, the largest experiment on human beings in the history of the world.
Hello, and welcome to the Independent Physicians Podcast. Our topic today is the Great mRNA and DNA Reset. And this is part one, the science. Our guest is Dr. Robert Malone, who's the inventor and skeptic of mRNA intervention technology and author of the recent book, Lies My Government Told Me. And I'll let him unwind his background for you. Thank you for being here, Dr. Malone.
Well, thanks so much, and thanks for a chance to talk to you today and to your audience. So, it's an auspicious day. Today is the 1-year anniversary of the Joe Rogan podcast that set the world on fire and changed my life. So it's a great day to be cutting a new video.
Who thought 3 hours of your time would result in millions of awakenings out there and people actually asking questions for a change? Thank you. So can you tell us a little bit about your background and training, say med school and thereafter, briefly?
My career goal was to be a retroviral vector-based gene therapy specialist for inborn errors of metabolism, pediatric genetic disease. Most of my truly rigorous scientific training occurred in my 3rd and 4th year of undergraduate when I was a lab rat. While I was in the laboratory, the discoveries were made about the role of a retrovirus in a non-human primate immunodeficiency syndrome. That was one of the key findings at the very earliest days of the AIDS cascade, let's call it.
So we're talking 1980s, just to set the time.
Yeah, that was 83, 84. So a lot of work was being done there at the time in vaccines. Then I really worked my can off. That's where I learned how to make RNA, purify from cells and tissues, and work with the molecule and do various types of analyses. And I could pretty much go wherever I wanted. The best gene therapy program in the world was at UC San Diego with Ted Friedman, who originally conceived of the idea. So I was accepted into UC San Diego with a focus on gene therapy and molecular biology. Convinced Inder to take me into a laboratory that was all postdocs. Just to a little more context, the senior postdoc in the lab, Dinko Valerio, created a company called Crucell based on technology that he was pioneering at the Salk when I was there in the lab. And Crucell got sold to Johnson & Johnson. That is the technology platform for the Janssen vaccine. So all of this roots back to the Salk Institute and Inder Verma's lab.
So you were right there at the pre-genesis of it all. That is to say, you were there on the first date.
Yeah, at a place that had half a dozen Nobel laureates plus Jonas Salk was still alive. I've met and had lunch with Francis Crick and many others. So that was a lot of lessons. As I was pursuing my research into how RNA is packaged into retroviruses so that I could make a more efficient manufacturing process for retroviral vectors, I had a series of discoveries that led to the ability to manufacture large quantities of highly pure mRNA synthetically in a test tube and deliver that into cells and then tissues. I left and joined a little startup across the street called Vical, brought my technology, protocols, reagents, etc. And within a couple of months, we had discoveries that led to a science paper. The first body of work was published in Proceedings of the National Academy of Science, is considered really the seminal paper in mRNA manufacturing and delivery.
Yeah, which is just, that's just absolutely critical stuff.
And built a gene therapy program with 3 other faculty and a number of students and postdocs, and then was recruited to UMaryland, Baltimore during that time at Davis. as well as a startup company called Inovio that still exists, that's based on other technology that I discovered for delivery, gene delivery.
Right, right. Inovio had a potential vaccine for all of this
Correct.
that went
Yeah, so
on and got
that's
sat on and squashed to death.
yeah, that's so that tech is originally came out of Norway, and my own laboratory. And when I joined Dyneport as associate director of clinical research for all of their biodefense products, which was virtually all biodefense products for the Department of Defense, so that's drugs, antibodies, and vaccines. Spent basically the next decade mastering regulatory affairs and clinical research, and during that time did a postdoctoral fellowship at Harvard University in global clinical research. So that covered regulatory affairs, clinical research, bioethics. I'd received bioethics training also from a bioethicist at U Maryland. Also worked for Solvay Pharmaceuticals. So I've worked for a big pharma as clinical director of influenza vaccines, and worked for ARIS Global TB Vaccine Foundation, which is one of the early Bill and Melinda Gates funded operations, and a number of others. And then I just hung up a shingle and started doing consulting, and I basically had my own small consulting shop for a number of years.
And where are we? 2010 at this point? Or
About then when I started doing the full-time consulting gig, and during that I've captured over, I think it's over 10 billion with a B dollars in grants and contracts, mostly contracts. Often been called to sit as study section chair for large vaccine contracts for NIH.
can I ask, just because of the timing, did you have knowledge or of any of this research that the government was doing in gain of function? Did you have any knowledge at that point, 2010 or 2012?
I have no touch points with these notorious characters that we've heard about, EcoHealth Alliance, DASIC, etc. Gain of function research has been threaded through the spaces that I've worked in, but I've never done that kind of work with virology or other pathogens. One of the senior scientists at UC Davis when I was there absolutely was doing gain of function work in trying to build a more pathogenic simian immunodeficiency virus by swapping in segments of the AIDS virus together with human cytokines.
Mm.
And I objected to that, raised concerns, and that's part of why I ended up leaving UC Davis. So I'm very
Were you
familiar.
labeled disruptive, doctor? Or
No, I was just quietly, opportunities that had been opened for me suddenly were no longer available.
so you saw them going in a direction that was unsavory to you, and you thought, well, I'll just take a different path.
More than unsavory, was absolutely unethical and inappropriate.
Okay, I don't mean to put words in your mouth. By all means.
High risk to insert human cytokines into a retrovirus chimera that included both simian sequences and human HIV sequences. So that's kind of my first touch point. And at that time, the standards were actually more rigorous for recombinant DNA back then than they are now, although there's this prohibition. But I have worked ever since I started working for Dyneport Vaccine Company and was kind of brought into the biodefense world. I have been granted from DoD secret level clearance. I have been exposed to information about what the government has been doing.
But one thing I wanted to get to, which is in your wheelhouse and no one else's wheelhouse, tell us about the use of mRNA and DNA with regard to having people make the spike protein in all of the tissues of their body, even across the blood-brain barrier.
So thanks for asking that. Early on, I spoke to the researcher at the University of British Columbia who has led the team, Peter Cullis, in the development of these current formulations. There were some advancements in the nature of the formulations and the compounds since Jill and I left the field, because we could not overcome the toxicity. And this is mid 90s, late 90s. So we abandoned the field because we just could not overcome the problems of the tech. Peter Cullis at
And
UBC.
problems of the tech, meaning that every time it was researched, the lab animals had problems, issues, couldn't get anywhere
Well,
near
that's one way to put
FDA
it.
approval. Is
We
that
we
correct?
we produced literally hundreds of different compounds and formulations and published extensively. That's another part of the press lies that we got out of the field and did nothing in just those two papers. And there's multiple. Behind me is a wall of patents that include many of those compounds, and some of which were marketed by Promega and others for research purposes. But I was being funded to pursue, in particular, cystic fibrosis. And because I had an appointment at the California Primate Research Center at Davis, I was able to arrange to get animals that would otherwise be sacrificed because they had come off study, control animals, etc., and be allowed to use them for pilot research having to do with polynucleotide delivery, DNA and RNA, into rhesus macaques using these lipids and this technology. And what we ran into again and again in both monkeys and mice is that these complexes are incredibly inflammatory. They provoke a massive acute polymorphonuclear infiltrate. And we just couldn't get around it. We were the first one. I was the first one to describe this. It basically killed the research program at Genzyme, which had been even more extensive than what we had done.
Did it kill the macaque monkeys?
Oh, more than kill them. Their lungs, for instance, just became a consolidated mass of acute inflammatory cells. They had to be sacrificed because of compromised pulmonary function. It was just not ethical to not sacrifice them because of the health impact caused by the administration of the product. So we got out of it. Peter Cullis had some advancements. So did Carrico and Weissman, involving the use of pseudouridine. And that gave rise to the current material. And I called Peter Cullis early on and asked him questions about the current formulations. For instance, why do they include cholesterol? Why do they include polyethylene glycol, et cetera? The field had come to believe that subtle modifications in the structure of the positively charged fat, the cationic lipid, would yield relative specificity in terms of organ targeting. Now, I believe that those conclusions, as I've looked back at the old literature, were really the consequence of biased investigations, because that was a goal that people were seeking, and relatively small numbers of animals. But that was the kind of belief system that existed in the field, that specific structures of these positively charged fat drugs would cause targeting or specific organ targeting. And in theory, early on, the logic that was trotted out to the public was that the specific formulation that was used would remain localized in the deltoid and the draining lymph nodes. It would not distribute to other tissues. But we know from the Pfizer common technical document, so that's what everyone was told. Docs like you were told the RNA would not stick around. It would be degraded rapidly like regular RNA. We now know that's false.
Because it's pseudo-uridinated mRNA, that it's been chemically changed, and therefore the body can't break it down like it breaks down its own message.
Yeah, that seems to be the case. And furthermore, the pseudouridine is intrinsically immunosuppressive, which is why it was used in this way, because of that problem that I mentioned of the hyperinflammatory response to these complexes. When Carrico and Weissman did those studies, the science of pseudouridine was just barely beginning to be identified and explored, and it still remains relatively immature. We don't really understand the mechanisms by which these things happen, the suppression of immune response, for instance, and the
May
regulation
I,
of the degradation.
if you don't mind, let me just simplify for folks because I like to put things in a really simple, straight way. And you being a physician who's a researcher and who's been in the field for 30 years, very matter of fact, very fact based and experience based. I just wanted to keep it straightforward for some of the listeners and correct me if I'm wrong at any time. So basically, what this is it's a piece of mRNA or DNA, which is a genetic message that's wrapped in a fatty globule, more or less, that allows it access into tissues where it wouldn't go otherwise and prevent it from being degraded until it gets into the cells. And, originally we were told it would be in the muscle cells, but now we know, I think it's like 17 or 19% or something stays in the muscle, and all the rest goes throughout the body. And because of the fatty nature of the globule that it's located in, it can cross into all kinds of highly metabolic tissues: ovaries, testicles, heart tissue, we know, where it causes a lot of side effects and inflammation. It also can cross the blood-brain barrier and can cause lots of degenerative neurologic disease potentially. So it gets into the cell, it injects or degrades, I should say. It degrades, and then the mRNA
It
or
actually fuses
DNA is
and
in there.
kind of distributes within the cell membrane. The fats do, and
Okay.
releases the RNA. At least that's the current model. Um, uh, but basically, you've got it right. The mRNA uh, is then in the cytoplasm, the white part of the fried egg, if you think of the yellow part being the nucleus. So it's in the cytoplasm where it is transported and loads onto ribosomes, which for the uninitiated non-molecular biologist are the little protein manufacturing machines that exist in your cells. And the RNA is kind of akin to a ticker tape. That's an old metaphor, but it's a good one.
It's a
It
messenger.
it contains the message, and it is like a ticker tape. It is read in a stepwise fashion by the ribosome as it transits this linear sequence of information and directs the production of protein. And it can direct the production of pretty much any protein you want. But in this
So
case, they chose to make it the spike protein of this particular virus.
and the spike protein, to my knowledge, is what is so inflammatory about COVID. I mean, they could have picked any protein, I guess, on the code of COVID-19. Instead, they picked the worst possible, most inflammatory weapon for your body to produce and then post on its own cells. And then your immune system sees those and goes, oh my God, I've been invaded. I'm going to have to kill my own cells now. I mean, is
Well,
that
that's true
oversimplified?
with any antigen, any foreign antigen. But spike itself is a toxin, and I was highly criticized for saying so. And some of the fact checkers who had no formal biology training back then would insist that there had been modifications to the spike protein designed to reduce its toxicity. That was absolutely false narrative. The two proline modifications that are made in the spike sequence are there to increase its immunogenicity. And the choice of this protein reflects the mad rush to get something out the door. It had been the
Whose
analogous.
idea was it to use this? I mean, is it the communist Chinese entity? Was it our own research people? Was it Pfizer? Was it Moderna? Do you
It
have any
absolutely
idea?
wasn't Pfizer. Pfizer. It's important to remember that Pfizer is a latecomer in this. The three early entrants were Moderna, really four early entrants. Moderna teamed with the NIAID's Vaccine Research Center. Uh, so that's, remember, Tony Fauci trotting out the team and emphasizing that one of the team members was African American, and speaking about how this should cause African Americans to want to take the vaccine.
Identity politics and marketing.
Right off the bat. And another one was called CureVac. CureVac did not use the pseudouridine. They're based in Europe. and another one was BioNTech, uh, and BioNTech was based in Germany.
Oh, they
And
were bought by Pfizer or
not
annexed by
not
Pfizer or
bought,
something?
not no, they have a marketing agreement, is a better way to put it.
Oh, okay. That
Sm-
happens in pharmaceuticals a
yeah,
lot where a small
small
company makes
biotech.
something and then a
Yes,
big company with a sales force says, "Hey, we're going to license this and we'll pay you for it, but it's going to be ours technically."
right, exactly.
Okay.
So that's why you see in the FDA licensing agreements uh, that the emergency use authorization is granted to Pfizer, but the license uh, that has never been used in the United States is granted to BioNTech. So
Gotcha, which
it's
makes
a subtle
sense.
nuance. Yeah. Um, so
So, go ahead. You were
so
saying.
we're talking about the rollout of these products and their genesis.
The facts
So
that they were rushed and they picked a pathway to use that wasn't necessarily ready for prime time. It had always been inflammatory, and
well,
they
and what had been the reason why this really was done, as near as I can discern, is that there had been a significant investment over time for vaccine development for both SARS-1 and MERS.
that's
The
20
middle,
years of time and money and
right,
interest.
and none of those vaccines succeeded. And in fact, I wrote an article together with Daryl Rickey in the beginning of 2020 that only made it to preprint servers, could not get it published, in which we talked about the history of vaccine development for coronaviruses, which repeatedly failed in large part due to antibody dependent enhancement, with a couple of exceptions of a couple of veterinary vaccines.
And I
Um,
think Dr. Fauci had said something about and one of the few times he was actually speaking accurately, he said there are some times where a vaccine can actually make you worse when you get the disease. And I think he was
yeah, that's an
referring
older
to
clip.
that.
And well, there's that. The most notorious example is the respiratory syncytial virus vaccine in the 1960s, in which and it's important to know that RSV was not in the human population uh, um, in the 40s, for example. The current evidence is that RSV entered the U.S. population, one of the leading theories, as a laboratory escape from a Bethesda, Maryland research laboratory, federally funded, that
Wow.
was doing non human primate research. So RSV is something that didn't used to be a problem until it escaped from a laboratory.
And
And
now we
there,
see it every winter and it's a
yeah,
problem for
yeah,
children.
it's global. A major cause of death in neonates, in particular. And so there was an effort to build an RSV vaccine that was a formalin inactivated product, and it looked good in the animal models, and they deployed it in clinical trials in young children. And when the data came in, the vaccinated children were much more likely to die than the
Hmm.
unvaccinated children. So ever since then, that has been one of the strongest cautionary notes in all of vaccinology.
So Dr. Malone, you're talking about, RSV and the possibility of a vaccine as a cautionary tale, and here we are flashing decades forward with the rush to produce an mRNA or DNA spike protein based COVID-19 miracle preventative answer, and the research that was done wasn't complete and it was put through via emergency use authorization and it didn't go through all of the usual FDA pathways that could take, 3 to 10 years in that case with regard to safety and efficacy. And then they said, well, this isn't really designed or being studied to prevent the disease or to prevent spread. It only minimizes symptoms, which I think is what they call low hanging fruit, I guess, with regard to that. And at what cost? Because there is a blank package insert with regard to inflammatory disease that might be caused either by the vaccine in and of itself or the vaccine after you get COVID and then potentially have that ADE phenomenon you were talking about.
So you put a lot in that sentence.
Sorry.
The ADE, um, although it was identified by the FDA as a risk and was identified in the literature as a major risk in coronavirus vaccine development historically, does not look like it's the major problem, although it may exist to some extent. What we're observing now in the present with Omicron, as opposed to whatever happened in the past in an environment in which all of our data are contaminated by a selection bias by the CDC and otherwise. So in the present, we now see, for instance, the Cleveland Clinic paper showing that the more inoculations you get, the higher risk you have of disease, hospitalized disease and death. We have information from all over the world indicating that there is a dose-dependent effect in this enhancement of risk of disease and death. And for those that have analyzed the timelines, it appears that these boosters provide a short-term bump in protection against these clinical outcomes, but that it has a slope, and that slope crosses zero protection or effectiveness at about 2 months after the booster inoculation, and proceeds to continue to become less effective and results in our patients having a greater risk of hospitalization and death after about 2 months of receipt of the booster product.
As opposed to not getting any of the boosters or not getting any of the shots.
Correct. It's the data are really contaminated now because we basically vaccinated the control group. Is an intentional strategy early on, but
well, an intentional strategy early on. Why?
That's the question that I think we would all love to see Tony Fauci forced to testify on. I can't get into his head, and I could only speculate, and I prefer not to. But
may I speculate? Is that okay?
You can say whatever you want.
Okay. Well,
but the data are the data. I mean,
right.
They did that.
Right. Well, my thought is that when they had the small groups that they tested, and then one group got, one group didn't get, and then they didn't have a great result, so they then said, well, it's an emergency, we have to give everybody the shot. Then you've got the group that got the shots, and then the group that didn't get the shots. Now everybody's got the shots. So anybody that has a side effect or problem from the vaccine would be assumed just to be general community or due to COVID or what have you. It wouldn't be attributable.
That is absolutely a valid interpretation. If we imagine that Tony Fauci is a nefarious liar, certainly having dealt with him and watched him my entire career, I can assure you I've seen multiple examples that the rules of clinical research do not apply to Tony Fauci. He will break the blind and disclose information and disrespect the normal boundaries in terms of maintaining integrity of control groups, etc. He's done it all the way through his career, and no one will question him because, of course, he controls all the money, or did until tomorrow.
So absolute power corrupts absolutely, whether it's science or medicine or name something.
Humans are humans.
Right.
So whatever the case, where I was trying to go with that is that when we do any analyses now, we face a complex epidemiologic landscape because so much of the population has been infected. And so you have a very complex landscape of not vaccinated but infected and recovered. We call those naturally immune. You have vaccinated then infected. You have infected then vaccinated. I'm an example of that. And all kinds of mixtures of the above. Now we have multiply vaccinated, multiply infected. But as you point out, there was never any attempt to demonstrate that the products would protect against infection, replication, or spread. And when I said that they don't protect in that way from Omicron on the steps of the Lincoln Memorial, just a little less than a year ago, in January of last year, or this year, I guess it is, I was called a liar for having said it. But that's now kind of conventional wisdom that they, as you say. And one of the things that I wanted to pick at in what you said in that very long sentence earlier,
Please.
is you were speaking as if the position that the products protected against disease and death was an intentional initial position. It wasn't. It was a limited hangout. As you'll look back and remember, over time, what we saw was an initial assessment insistence that if 70% of the population would take these vaccines, we would achieve herd immunity, meaning there would not be replication or spread.
And herd immunity before that had been due to an amount of the population being naturally exposed and developing immunity. Is that not a correct definition?
Herd immunity can be achieved in a number of different ways. It could be achieved by a vaccination that was sufficiently effective in theory.
Okay, so it could be a combination of things.
But it was never achieved. And they had no data. Tony Fauci eventually was forced to acknowledge that he pulled those numbers out of a hat or whatever dark place. And you'll recall that he gradually he ratcheted upwards the percent of the population that he insisted would have to take the product. So the same thing happened in Canada under Justin Trudeau, the noted virologist and immunologist.
Right.
Who rumor has his family is invested in the company that was created by Peter Cullis at University of British Columbia. Just good to know. That has not been verified. But Fauci and really, we now know the whole crew depended on, the term that was used by both Rochelle Walensky and Deborah Birx is that they didn't know that these things would work. They hoped that they would work. So now in retrospect, we see that these enormously damaging public policies, including mask use, particularly damaging for young children, lockdowns, and the vaccine products and the suppression of early treatment were all based on hope rather than science. So these very egregious public health policies were based on basically seat of the pants hope by public health bureaucrats that really were not trained epidemiologists. You can recall the authors of the Great Barrington Declaration were disparaged by Tony Fauci.
Sure.
When in fact they were full professors at 3 of the top universities in the world in epidemiology. Tony Fauci is absolutely not trained as an epidemiologist. Neither is Deborah Birx. Deborah Birx, however, is a Tony Fauci postdoc. So there's that.
The 6 degrees of Tony Fauci in the government.
Well, Tony Fauci, remember I mentioned Carrico and Weissman.
Okay.
Drew Weissman is another Tony Fauci postdoc.
Gotcha. So let me just finish up with telling us a little bit about the potential damage to an immune system that one of these products that's currently available and being pushed in radio, TV, film, and
the
internet
tune of
ads.
475 million.
Yes.
Of your tax dollars.
Tell me and tell our listeners, if you would, about the potential damages. I mean, we hear about pericarditis in young people, but in my practice, I've seen a wide variety of neurologic, cardiovascular, cancer,
Stroke,
and cancer recurrence issues. Maybe it's a question for Dr. Ryan Cole, but from your perspective.
Immunologically, we have multiple pathways for damage. The one that I've highlighted when I testified in the Senate in Texas, sworn testimony to the Texas Senate a number of months ago, was the problem of what's known immunologically as original antigenic sin, something else I've talked about all the way through this as a risk, otherwise known as immune imprinting. And there are multiple papers from top laboratories in the world, including this notorious Cell paper that I often cite from Stanford of February of this year, that was the one that showed by lymph node biopsy that the mRNA persists for at least 60 days, if not longer, in human recipients.
So not 28 seconds, but 60 days at the least.
That's all they tested, and also showed that the level of spike protein produced after these products are administered in the plasma, in the blood, is significantly higher than the levels produced from natural infection. So that addresses the why do we see more problems with the jab than we see with the natural infection, because they both produce spike. The answer is a lot more spike, and the kinetics of that, it comes on really fast after the injection and then tapers slowly. That's absolutely not the kinetics of the natural infection, where it gradually builds and then it gradually tapers.
So if I may generalize, so the shots, the mRNA and DNA shots, produce a certain level of inflammation and potential disease that's above that of the natural infection of COVID. What happens when someone takes the shots and then gets COVID, or someone has had COVID and then forced to take the shots?
So that's a great lead in. So there's a super paper came out of the UK, among others, that documented this problem of immune imprinting. And I spoke at length, gave multiple lectures, wrote multiple essays about this problem, warning people not to take the boosters, because they, in my estimation, are perfectly designed to exacerbate the immune imprinting problem. So what is immune imprinting or original antigenic sin? Your body's immune response is biased by what it has encountered in the past. And to go into the mechanism of how that happens in terms of the immune response is, let's say, too technical for right now.
You don't have to go into details. Just give us the generalities based on your experience and
the literature.
Knowledge.
It's literature from top laboratories in the world, huge research groups. What the data show is that both with influenza vaccines, matter of fact, this is the problem with the repeated annual flu updates, is this same immune imprinting problem. Your body learns from its initial exposure to an antigen, and it retains that as memory. And then when it's re-exposed to a related antigen, it responds as if it's the first one that it encountered. And it's more difficult for it to switch to the specificity of the new version. And in a rapidly evolving RNA virus, like flu, like a coronavirus, this creates a bias problem. Prepared for the last war, not for the current one. And that introduces bias in how it's responding. And in the case of these boosters, what is being done is to drive your immune response further down that road of bias. You're reinforcing the bias.
That is to say, bias toward more inflammation or less inflammation and tolerance?
Bias towards antigens that are no longer in circulation because the virus has evolved. So you're
saying
driving,
old shot, new disease.
that's one way to think about it. But now there's a new paper out that's circulating quite a bit that goes even deeper into this and looks at the subcategories of immunoglobulin, of antibodies that are being produced. And it appears that with these multiple inoculations, which we know are associated with increasing risk of disease and death, right? In addition to the increased risk of the toxicity that's intrinsic in the formulation, right? Myocarditis, et cetera. Immunologically, it's driving your immune response towards an IG4 antibody. And an IG4 antibody subtype is typically associated with allergy responses, and has very different functional properties from the normal immunoglobulins that are produced in response to an infection. Mm-hmm. So it appears, based on this one paper recently published, that part of the mechanism for this immune imprinting bias that's occurring, wherein the more you jab, the less effective they are, um, uh, is that you're actually pushing the immune system down a road that biases towards the type of antibody response you would see in an allergy response, which is not an effective antibody type for infectious disease viruses.
Well, that's interesting because that kind of goes to my last question with regard to this, which is, so despite the shot being dangerous and potentially causing harm to citizenry, why not keep changing the shot and keep doing it every 2 months and what have you? So what I hear you saying is, I mean, you learn that in counseling, right? Whether you're the counselor or the counselee, that when they keep changing the shot and things, and the virus constantly is changing and wobbling and changing its configurations and things, that there's constantly inflammation that's going on and there's risk to people, and the virus is constantly trying to change away from that, just as a tinkerer that it is. And you'll never hit it, number one. Number two is you're constantly going to potentially burn out segments of the immune response to things because you're
I think that's a good way to
hyperstimulating.
think about it. I think it's a good way to think about it. Burning out immune response is a simple metaphor that we can all understand. So
Thank you.
good for you on that. And it's worse than that because the intrinsic toxicities of spike and its interaction with the nucleus, with mitochondria, and some of the activities of the complexes themselves seem to be suppressing adaptive immune responses, particularly cellular immune responses, which may explain the elevated risk of shingles, Epstein-Barr virus, cytomegalovirus, etc., reactivation that was early on known as one of the risks associated with the vaccine inoculations. So, and that, of course, feeds right into this uh, um, third wave risk, let's call it, of cancer. Because cancer, as you know, is always arising in our bodies. The notorious example is if you and I think we both identify as male. I certainly do. And whether we identify as male or not, we both have
Biologically
prostates.
male as well.
But we both have
Yeah,
prostates.
right.
And if we live long enough, we will die with prostate cancer. It is not necessarily clinically significant prostate cancer, but it will happen. We have cancer arising in our bodies all the time is the point, and it's particularly our cellular immune response that keeps it in check. And so these odd phenomena of reactivation of latent DNA viruses like shingles, and this now
Like chicken
growing.
pox is living in our system and our body's keeping it under wraps as long as the immune system is competent. But when you poke the bear with these mRNA and DNA shots, the gel cell opens and then you get reactivation of what you're saying, all the latent viruses.
And in what's concerning, well, I'm going to choose that word, about the oncology reports that Ryan Cole and now pathologists and increasingly
Dr.
oncologists are
Ray
reporting,
Page as well.
um, is that we're observing these characteristics of not only reactivation of what we thought was suppressed cancers. Um, we're seeing the as Brian says, the cells don't lie. We're seeing very high numbers of mitoses per field. That means the cells are dividing very fast in many of these, much more fast than would normally be the case with those types of cancers. So this suggests that we're having both a suppression of the ability of the immune system to control the cancer, as well as some other phenomena that is altering the fundamental biology of those cancers. And this may relate to toxicities associated with spike itself. So that's a wide open area in terms of research. And it's a controversial area right now, talking about these potential oncology risks. Um, as you know, cancer tends to develop and present slowly. And it's only over time that we observe these phenomena and build up enough data that we can say, oh, now we got a problem. But there's been this massive rush to inoculate the world, uh, and and
With
without
no
any
control group,
with
no
no control
control
group,
country even.
and no solid information about long-term risks, because of course we can't have that information. Anybody here in the audience knows that we can't know about what's going to happen 5 years after inoculation, because we only have had a year and a half of data.
Oh, for sure.
So there's no way we can reassure our patients
Can I
that
mention
there's long-term
a quick
safety.
analogy that I found useful? When I talk about the immune system, a simple way to understand it is there's different things that it does in different parts, and it's quite complicated, as you know from your career. However, I tend to simplify for patients. I used to say, well, there's kind of two major goals of the immune system. One is the army or the military, that is to say, to keep things out. And it goes from, the skin to the other cells and things that physically keep things out. And if something tries to get in, it tries to keep it out. The other function is surveillance. That is to say, to your point, when cancer cells come up in us every minute of every day, that our body picks them out and kills them and takes them off to be shredded, off to the shredding room. Having said that, when something goes in and pokes the bear that's got everything under control and got complete surveillance, and it constantly pokes it or forces it into an unstable state, which I believe the mRNA and DNA vaccines do, lots of bad things happen, that the body can't defend itself against infectious disease. And sadly, the body also can't pick out all of the cancer cells that it would normally get rid of and prevent a disease.
And to add another log to your fire, um, as
Please.
you have identified, we're also seeing signs of autoimmune mediated neurologic disease, and this odd new clinical syndrome of thrombotic thrombocytopenic purpura. This we've known about TTP, but we're seeing a very different form. And it appears that part of what may be happening in the autoimmune disease part is also a dysregulation, but it may also be because these particles are really sticky, and spike protein itself is sticky.
Mm.
And so they are adhering to natural proteins, which your immune system apparently is observing and responding to as a hybrid protein, of a mixture of your normal protein and this foreign protein or other particulates, these lipid nanoparticles. And that's a well known pathway to break tolerance against self. We call this use of haptens. And so it appears that something like that is also occurring and is leading to a subset of patients developing a variety of autoimmune diseases, including neurologic ones that you've mentioned, as well as blood problems having to do with blood clotting on top of
So
all the other
more
blood clotting.
mRNA and DNA interventions at this time, worse prognosis, less mRNA and DNA interventions, or zero, better prognosis. Is that a fair generalization across the data?
I believe it is. And the FDA is in complete denial of this. And currently, there's over 200 clinical trials for new mRNA complex based medicines, and over 50 of them currently enrolling.
Mm.
And the FDA has
Wow.
taken the position that was discussed at the UN quite a while ago, that these current manufacturers of this material, which is purported to be safe and effective by the FDA and the CDC, will be allowed to substitute in new RNAs for the existing spike RNA,
Mm.
basically at liberty, with very little additional testing, and proceed into human clinical trials based on the thesis
So
that
they're going to shorten the shortened process.
they have already. that's already accepted, and they're
Wow.
fast tracking some of these products. And um, there's data that I've seen that was disclosed, for instance, by Moderna in one of their board meetings, not to public, having to do with their new influenza product, that demonstrates that at the doses currently being used for the COVID vaccines, their influenza vaccine product, which does not have spike, was having serious adverse events in the rate of about 30% in phase 1 trials.
Wow.
We wrote an
Right.
essay about
Wow.
this. So what this demonstrates is that it's not just the spike, and it is the formulations itself.
It's the formulation, it's the mechanism, the not ready for prime time players, if you will. Can I also ask, because we could talk for hours and hours, and
How else can someone get a hold of you or read your Substack? What do you think?
So thank you for that pump.
Welcome.
The Substack essays are written by Jill and myself, about 50-50 on a daily basis. That's rwmalonemd.substack.com. I'm also on Twitter. I'm right at the threshold of 900,000 followers. Yay. Hoping to hit 1 million. So at rwmalonemd. Also on Getter, Gab, and Truth Social. And the Malone Institute, maloneinstitute.org, is also where we put the longer documents, like, for instance, the spreadsheet that lists all the most comprehensive spreadsheet of all World Economic Forum trained young leaders. That is, you can download it in Excel format and search by industry, by name, by nation state, by year graduated. It's a 5-year training program. And we have many other documents there that you can find and make your own exploration, your own decisions. So that's maloneinstitute.org.
So that'll wrap it up for this edition of the Great mRNA DNA Reset for the Independent Physicians Podcast. I'm your host, Dr. Craig Wax. We'll see you next time with another excellent expert and another thoroughly important topic right here. Thank you. Here's your fist bump. Be well.